The Green Empire, Amazon's Lost Cities and the DNA Truth. LiDAR surveys and DNA analysis found, when researchers applied them systematically to the Amazon basin beginning in the 2000s, evidence of a civilization so large, so complex, and so thoroughly erased from the Western historical imagination that its rediscovery required not just new technology, but a fundamental revision of what scientists had assumed was possible in tropical rainforest environments. The story begins with a legend. El Dorado, the Gilded King, the Golden City, was the obsession of Spanish conquistadors from the moment they arrived in South America in the early 16th century. Indigenous people they encountered described great settlements to the east and south, cities of wealth and ceremony that lay beyond the rivers and the forest. The Spanish pursued these accounts for over a century, sending expedition after expedition into the Amazon basin, and finding, to their reported bewilderment, nothing but trees. In 1542, a Dominican friar named Gaspar de Carvajal accompanied the first recorded European navigation of the full length of the Amazon River. His journal described populous settlements along the riverbanks, towns that stretched for miles, ceramic-covered shores, roads, cultivated fields, and people in the hundreds of thousands. His account was dismissed for centuries as fantasy, the exaggeration of a man who had survived an extraordinary ordeal and whose credibility was questionable. It is now understood to be among the most accurate early records of what the Amazon actually looked like before European disease destroyed it. The last major figure in the tradition of Amazon exploration was the British Army officer Percy Fawcett, who disappeared in 1925 while searching for what he called the City of Zed, a lost civilization he believed lay hidden in the Mato Grosso region of Brazil. He was never found. His disappearance became one of the 20th century's most famous unsolved mysteries, and the city he sought is no longer a fantasy. Remote sensing data found, when LiDAR, light detection and ranging, was applied to large sections of the Amazon basin from aircraft beginning around 2010, evidence of urban infrastructure on a scale that the scientific community had not anticipated, and that required multiple independent confirmations before the results were accepted as valid. LiDAR works by firing rapid laser pulses from an aircraft toward the ground and measuring the return time of each pulse. In a forest, some pulses return from the canopy, some from mid-level vegetation, and some penetrate all the way to the ground surface. Computer processing separates the ground-level returns from the vegetation returns, producing a precise three-dimensional map of the terrain beneath the forest cover. Features that are completely invisible from the air or from the ground, earthworks, roads, building platforms, canals, appear on LiDAR maps with the clarity of a surveyor's drawing. What LiDAR revealed in the Llanos de Mojos region of present-day Bolivia, in surveys published in the journal Nature in 2022 by a team led by Heiko Prumers of the German Archaeological Institute, was a network of urban settlements belonging to what archaeologists now call the Kasarabe culture, dating roughly from 500 to 1400 CE. The largest sites covered areas comparable to medieval London, connected by elevated causeways, some straight for over 10 kilometers, and by canals that managed seasonal flooding. The settlements included conical platform mounds reaching 22 meters and U-shaped ceremonial structures built to precise orientations, supporting populations the researchers estimated in the tens of thousands at the largest centers. This was not a dispersed hunter-gatherer population living lightly in the forest. It was a managed, engineered landscape, a civilization that had transformed the Amazonian environment on a continental scale and that vanished from scientific awareness so completely that its rediscovery required technology that did not exist until the 21st century. Geochemical analysis found, when researchers examined the dark, extraordinarily fertile soil known as Terra Preta do Indio, Amazonian dark earth, that this material, distributed across hundreds of sites throughout the Amazon basin in deposits that can be meters deep, was not a natural geological formation, but a human creation. A deliberate soil amendment technology developed over centuries that produced results no modern agricultural chemistry has been able to fully replicate. Standard Amazonian soil is oxisol, heavily leached, low in nutrients, poorly suited to intensive agriculture. The tropical conditions that produce the forest's biological abundance simultaneously strip the soil of the mineral and organic components that support sustained cultivation. How any large pre-Columbian population fed itself in this environment was for decades considered one of the central arguments against the existence of complex Amazon civilizations. If the soil could not support intensive agriculture, the cities could not exist. Terra Preta resolves this paradox. Its composition, analyzed by archaeologists including Bruno Glaser of the University of Bayreuth and a generation of subsequent researchers, includes high concentrations of charcoal in a form called biochar, which resists decomposition and provides a stable carbon matrix. Combined with biological material, including bone fragments, pottery sherds, and organic waste, and populated by specific microbial communities that continue to enrich the soil centuries after initial creation, Terra Preta is self-renewing. Sites in the Amazon where human communities created Terra Preta 2,000 years ago remain among the most productive agricultural soils in the region today. The technology required systematic effort over generations, not a single innovation, but a cumulative practice of soil management that implies a level of ecological knowledge and long-term landscape planning inconsistent with the simple hunter-gatherer model that dominated Amazon archaeology until recently. DNA research, published in the journal Nature in 2015 by a team led by David Reich at Harvard Medical School, found, when they analyzed the genomes of indigenous Amazonian populations, including the Surui and Caritiana peoples of Brazil, a genetic signal that was not present in any other Native American population, and that pointed toward an unexpected and still not fully explained connection between pre-Columbian Amazonia and the populations of Australasia. The signal, identified in the study as Population Y for the Yipiquera, meaning ancestor in the Tupi-Guarani language family, consisted of genetic variants found in present-day Aboriginal Australians, Papuans, and Andaman Islanders that were absent from all other Native American groups tested. The statistical analysis showed that the Amazonian populations carrying this signal shared more genetic material with Australasian populations than could be explained by random variation or analytical error. The finding was replicated in subsequent studies and has not been successfully explained by any simple model of human migration. The standard model of the peopling of the Americas involves a migration from Northeast Asia across the Bering Land Bridge approximately 15,000 years ago, with subsequent diversification within the Americas. Population Y does not fit that model. It requires either a separate migration event that brought Australasian-related people to South America by a different route, possibly by sea, or a deeply ancient shared ancestral population in Asia that subsequently diversified in ways that the fossil and genetic records have not yet captured. Ancient DNA from skeletal material recovered from Brazilian cave sites has confirmed the presence of the population Y signal in individuals dating to approximately 10,000 years ago, establishing that this genetic ancestry was present in Amazonia well before European contact and was not a product of historical admixture. Archaeological evidence found at burial and ceremonial sites across the Amazon basin documents cultural practices that connect the region's pre-Columbian civilizations to the broader traditions of complex societies in the Americas, and that carry a biological record accessible to modern DNA and isotope analysis. Secondary burial in large ceramic urns, a practice documented at multiple sites across the Brazilian and Colombian Amazon, involved the initial burial of the deceased, followed after a period of decomposition by the exhumation of the bones, their cleaning, and their redeposition in elaborately painted ceramic vessels. The urns, recovered from sites in the Marajo Island region at the mouth of the Amazon, are among the most technically accomplished ceramics produced anywhere in the pre-Columbian Americas, decorated with complex geometric designs and human figures in styles that took researchers years to interpret. The bones within these urns are exceptional sources for ancient DNA analysis. Protected from the soil environment by the ceramic container, and frequently accompanied by organic materials including plant remains and worked shell, they preserve genetic material in conditions far superior to open-ground burial in the acidic Amazonian soil. DNA, extracted from urn burial remains at sites in Brazil and Colombia, has confirmed the genetic diversity of Amazon populations, and in some cases produced the population Y signal that links specific individuals to the Australasian ancestry identified in living populations. Isotope analysis of the same skeletal material documents diets that were substantially more varied and nutritionally rich than the forest environment alone would suggest. Evidence of the managed agroforestry systems that supplemented wild food resources with cultivated plants whose distribution throughout the Amazon is itself a record of human activity. Paleoepidemiological and demographic research found in the historical, genetic, and archaeological record of the Amazon basin documents what researchers now describe as the largest and most rapid population collapse in documented human history. An event whose scale was so enormous that it effectively erased the civilization Gaspar de Carvajal described within a century of his journey down the Amazon. When Francisco de Orellana navigated the Amazon in 1542, the riverbanks were populated and the settlements he described were active. When subsequent European expeditions traveled the same route in the late 16th and 17th centuries, they found the same riverbanks largely empty. The people had not moved, they had died. The mechanism was epidemic disease. Indigenous Amazonian populations had no prior exposure to the pathogens Europeans carried: smallpox, measles, influenza, typhus. Mortality rates in affected communities reached levels no modern epidemic has approached. Disease typically moved ahead of direct European contact, spreading through trade networks so that populations were dying before they ever saw a European. Estimates of the pre-Columbian Amazon population range from 10 to 50 million people. By 1600, roughly 90% were gone. By the time systematic European exploration of the interior began in the 19th century, the forest had been reclaiming the earthworks, causeways, and cultivated landscapes for 200 years. Ancient DNA, extracted from pre-contact and post-contact skeletal material from Amazonian sites, documents the genetic bottleneck associated with this collapse. A sharp reduction in genetic diversity at the population level that corresponds precisely to the period of European contact, and that is one of the clearest biological signatures of demographic catastrophe in the archaeological record. Rock art found at the site known as Serranía de la Lindosa in the Colombian Amazon, documented and published by researchers from the University of Exeter and the Universidad Nacional de Colombia in 2020 and 2021, constitutes one of the most significant archaeological discoveries in the Americas in the past century. A painted panel stretching approximately 13 kilometers along a cliff face depicting animals, human figures, and geometric designs in a scale and density that has no parallel in South American rock art. The paintings, executed primarily in red ochre, an iron oxide pigment that resists chemical degradation over millennia, include representations of animals that are not present in the Amazon today and that have not been present for approximately 10,000 years. Mastodons, giant ground sloths, paleolama, a large extinct camelid, and what appear to be giant armadillos are depicted alongside the deer, tapirs, and fish that continue to inhabit the region. The presence of extinct megafauna in the paintings establishes that humans were present in the Colombian Amazon and were creating representational art during or shortly after the Pleistocene, more than 12,000 years ago. The implications for the timeline of Amazon habitation are significant. The standard account of human arrival in South America placed the date at approximately 13 to 15,000 years ago, based on the oldest well-dated archaeological sites. The Serranía de la Lindosa paintings are consistent with this timeline and extend the documented record of cultural activity in the Amazon significantly earlier than most researchers had assumed. Ancient DNA from skeletal material of comparable antiquity from cave sites in Brazil has confirmed human presence at these dates and has shown that the earliest Amazonian populations were genetically distinct from later populations, suggesting multiple waves of migration and complex demographic history over more than 12 millennia. Ethnobotanical and genetic research conducted over the past 3 decades by researchers including William Millikan of the Royal Botanic Gardens at Kew and indigenous knowledge specialists working with Amazonian communities has documented a pharmacopoeia of medicinal plant knowledge whose depth and sophistication is only now being systematically evaluated using modern biochemical methods. The Amazon basin contains an estimated 10% of all plant species on Earth, and indigenous communities across the region have developed over thousands of years of empirical experimentation, detailed knowledge of the therapeutic properties of hundreds of species. This knowledge, encoded in oral tradition, transmitted through apprenticeship to shamanic practitioners, and applied in the treatment of conditions ranging from wound infection to neurological disorders, represents a biological database of enormous potential value that is simultaneously at serious risk of permanent loss as Amazonian communities face land dispossession, forced assimilation, and the deaths of older knowledge holders without successors. DNA analysis has contributed to this research in 2 ways. First, genetic analysis of cultivated and semi-cultivated plant species has documented the extent to which Amazonian peoples shaped the botanical composition of what appears to be wild forest, identifying dozens of species whose current distribution reflects centuries or millennia of deliberate human cultivation and dispersal. Second, pharmacogenetic research comparing the genomes of Amazonian indigenous populations with their documented patterns of medicinal plant use has identified genetic variants associated with drug metabolism that may explain why specific plant compounds are used differently across different communities. The relationship between ancient population genetics and pharmacological knowledge is an emerging research area whose implications for modern medicine are substantial and largely unexplored. Satellite imagery, aerial surveys, and limited ground-level contact, documented by Brazil's Indigenous Affairs Agency, FUNAI, have confirmed the existence of approximately 100 groups of people in the Amazon basin who have no regular contact with the outside world. Communities whose existence is known, but whose territories are legally protected, and whose isolation is maintained as official Brazilian government policy. The policy of non-contact, formalized in Brazilian law in the 1980s under the leadership of the indigenist sertanista Sidney Possuelo, following decades of disastrous attempts at forced integration, rests on a straightforward epidemiological reality. Uncontacted populations have no immunological experience with the common infectious diseases of the industrialized world, and contact, even brief, apparently benign contact, can trigger epidemics with mortality rates comparable to those that destroyed the Amazon's pre-Columbian civilizations 5 centuries ago. The risk is not theoretical. Documented cases of contact-related epidemics in the 20th century killed majorities of the affected populations within months. A common cold, introduced by a well-meaning researcher, can become a fatal respiratory infection in a community with no prior exposure to rhinovirus. The genetic data from contacted indigenous Amazonian populations is relevant to understanding who the uncontacted groups are and where they fit in the broader history of the basin. DNA studies of groups including the Yanomami, Awa, and Karubo have established deep genetic diversity and long population histories in their respective territories. The uncontacted groups, based on their geographic locations and the languages of neighboring contacted communities, are almost certainly the descendants of populations that survived the colonial-era collapse by retreating into the most inaccessible interior regions. The final living continuity with the civilization that the LiDAR surveys are recovering from beneath the forest floor. Scientific synthesis found across the LiDAR survey literature, ancient DNA research, and archaeological publications of the past decade presents a picture of pre-Columbian Amazonia that is, in almost every measurable dimension, the opposite of the image that dominated Western science for most of the 20th century. The Amazon was not a pristine wilderness untouched by human hands. It was a managed landscape, shaped over millennia by populations in the millions. Populations who engineered their soils, cultivated their forests, built their cities, connected their settlements with roads and canals, and developed bodies of botanical and medical knowledge whose sophistication modern science is only beginning to appreciate. The forest that European explorers and later scientists described as primeval nature was, in significant part, a cultural artifact. The overgrown remnant of a civilization that had been destroyed so thoroughly and so recently that the jungle had not yet finished reclaiming it. The DNA evidence has been essential to this revision in two ways. Population genetics has established the deep antiquity of human habitation in the Amazon, the extraordinary genetic diversity of its pre-Columbian populations, and the catastrophic demographic impact of European contact. Ancient DNA from skeletal and sedimentary sources has connected specific archaeological sites to the genetic lineages of living indigenous communities, giving those communities a documented biological continuity with the civilization the LiDAR surveys are mapping. What remains unknown is larger than what is known. The LiDAR coverage of the Amazon basin is still a small fraction of the total area. The ancient DNA record is fragmentary. The uncontacted groups carry knowledge and biological heritage that cannot be accessed without risk of destroying them. The Amazon's lost civilization is not fully found. It is in the process of being found. One laser pulse and one genetic sequence at a time.